Okay, so the study in this video is by Casey et al. So remember, more than 3 researchers, it's et al. And it's done in 2011. Okay, so as you can see from the pictures on the slide, it's looking at brain structures. But you can also see on that slide, we've got a picture of some marshmallows. So this study is looking at something called delayed gratification. Okay, so delayed gratification is about if you can wait for something in order to get a big reward later on. So if you are quite good at saving up your money, so instead of like going to McDonald's for lunch or treating yourself to a new handbag or to a dress and things like that every day or every week, you don't spend that money and you save that money up, and then you buy yourself something really good like a ticket to a festival or a holiday. Okay, so if you're quite good at doing that, you're quite good at delaying your gratification. So instead of looking for immediate gratification, which would be buying the reward right now, you save up the money and you delay that reward till later on when you can get a bigger reward. Okay, so what this study is looking at is the neural correlates of delayed gratification. So it's looking at if there is a specific part of your brain that is related to whether you can or cannot delay your gratification. Okay, so we need to think about why in this study we refuse to give in to temptation or why we give in to temptation. Okay, now the idea is that you have an ability to resist temptation in favour of long-term goals, and that is essential for individual, societal and economic success. So in other words, if you don't forgo the idea of going out and partying and getting all that pleasure, all that gratification now from socialising and drinking and all the other things, then you won't reach your long-term goals. So if you go to university and you spend all your time with your friends and going out, you're not going to get that degree in the end. Whereas sometimes you might think, well, I've been out this week, I'd better stay in for this weekend and study because I've got an assignment due in. And without that assignment, you're not going to get that degree. Okay, so you're delaying your gratification because you're thinking, well, once I've done the assignment, I'll go out next weekend instead. Okay, so you're delaying that gratification and I'll have a really good time next weekend, but this weekend I need to do this first. Okay, and same for like things like economic success. If you're spending all of your money straight away, you're never going to save up, therefore you're never going to be able to buy a house or buy a car or anything else that would make you an economic success. Okay, so the idea is that alluring situations can diminish your control, and what serves as an alluring situation that reduces the capacity to control your impulses changes as a function of age. So in other words, when you are quite young, what you see as alluring, so what is quite tempting to you, so whenever we talk about alluring, we mean tempting. When you're a child, something that's quite tempting to you is probably not tempting to you as an adult. So I could put a marshmallow or a cookie in front of a very small child and they would find that quite hard to resist. Whereas you, as teenagers, if I put a marshmallow in front of you and said, just wait 10 minutes, and if you've still not eaten it, I'll give you another one, that's probably quite easy for you to do. Because what you find tempting now is probably very different. Okay, now your ability to delay gratification depends on your cognitive control. So sometimes we have coping strategies, cognitive strategies that we use to help us delay gratification. Okay, and there appears to be these differences in between the strategies that individuals use. Okay, so it might be like distracting yourself or looking away, things like that. So I'm going to play this little video and I'll play it again in lesson. Actually, no, I'll just play it in lesson. Oh no, there we go. So I'll play this little video and it's a great video about delayed gratification. Okay, so you can see some of the things that the kids are doing to prevent them from actually eating the marshmallow, so just having a taste, looking away. Okay, so you can see particularly at the end of that video, like how it's quite difficult for a child to, resist that temptation, for a child to delay gratification. Okay, so like you saw the boy right at the end there, where is he? Really struggling, nearly putting it in his mouth and then stopping. So it's really difficult for a small child, whereas it's probably quite easy for you as an adult. Okay, so the idea is that you've got hot and cool stimuli. Okay, so all this is still going in the background. There's quite a lot of background information for this study. So you've got hot and cool stimuli. So cool stimuli are things which we find to be quite neutral. So they're not appealing, but they're not unappealing. We don't feel anything towards them. Okay, whereas hot stimuli are things that are appealing and tempting to us. So the look of the marshmallow, the texture of the marshmallow, the smell, the taste. They would all be hot stimuli. Okay, now one way in which people make themselves able to resist temptation is by cooling. So instead of looking at the marshmallow, we don't think of its hot properties like its taste and its texture and its smell, but we might think about the cold properties like its shape, or we might liken it to something else. So picture it like a cloud rather than like a marshmallow. Okay, and that's known as cooling. So that's just an example of one of these cognitive strategies that we can use to resist temptation. Okay, so the theory is, as we already know from other studies we've looked at, that parts of your brain are related to particular things. So some studies have demonstrated that a part of your brain called the inferior frontal gyrus is crucial in interpreting facial expressions and determining emotional responses to that expression. So if you see somebody walking towards you and they look really angry, your emotional response might be to feel a bit scared. Okay, so it's your inferior frontal gyrus that's helping you determine that emotion, so interpret that expression and also determine the response. There's also a correlation between avoiding risky behaviour and greater excitation in that inferior frontal gyrus. So if you scan somebody's brain while they're avoiding a risky behaviour, so they're not engaging in a risk-taking behaviour, that area of the brain appears to be quite active. So that suggests that perhaps that area of the brain is making that decision to avoid that risk. Okay, so to do with emotions and risk and making decisions. Okay, now if we look at these pictures, I want you to just think about how you feel. Okay, and same with these ones. Okay, there's such a thing known as emotional contagion. So it's kind of looking at how emotions can spread from one person to another. So when you see somebody smiling or somebody happy, you feel happy. If you see one of your friends crying and upset, you're going to feel upset, and you might feel like you're going to cry, even though it's not really affecting you, but you're upset because they're upset. And same when you're sort of with a group of friends and you might be like giddy friends and then you start getting really giddy even though you're not a giddy person. And the same thing when it happens to come across things like riots and football hooliganism and things like that. A group of people can affect your behaviour and other people can affect your behaviour, and that's to do with that emotional contagion, which is related to that inferior frontal gyrus, so determining the correct response. So if you with your friends and they're crying, the correct response that inferior frontal gyrus determines is that that's really upsetting, and then you feel upset. Okay, so you've got some different brain systems involved in delaying gratification. Okay, so Metcalfe and Mischel propose that there's cool systems and hot systems in the brain when it comes to self-control. So cool systems are to do with cognitive control. Okay, so for example, your inferior frontal gyrus would be a cool system. It's found in your prefrontal cortex. So it is responsible for controlling those thoughts, controlling your emotions, making decisions, resisting temptation. So it's quite logical and related to control. So it's a cool system. And then you've got hot systems. So hot systems are related to desires and emotions. Okay, so there might be emotional brain regions. These regions lack control. They might be seeking rewards. And these are more active when you're faced with tempting stimuli. So you've got an area in your brain which forms part of your limbic system called the ventral striatum, and that's a hot system, so it's seeking out rewards. It's quite a primitive emotional part of the brain. Okay, so there's a little diagram of where some of these parts are. Okay, so the ventral striatum, so this is your limbic system here. So all these sort of coloured parts here are your limbic system. Okay, so within this you can find your ventral striatum. Okay, so there we go, there's your ventral striatum in that limbic system. So it's been found that the ventral striatum facilitates and balances motivation with higher and lower level functions, okay, such as inhibiting one's behaviour in a complex social situation. This region has been found to be in the region of the basal ganglia neural circuit, most associated with rewards. Okay, so you can see there that this blue structure is your basal ganglia. Okay, and part of that basal ganglia, there we go, there's your basal ganglia, this time in purple. Part of that basal ganglia there is your ventral striatum. Okay, and your basal ganglia, that neural circuit is to do with getting rewards. So we think that that ventral striatum must be related to reward seeking, perhaps instant gratification. Okay, so if we look at brain images, they provide evidence for dissociable brain systems related to immediate over long-term choice behaviour. So dissociable essentially meaning separate. So you've got separate brain systems for getting immediate gratification, and then you've got another separate brain system for long-term choice behaviour, perhaps delaying gratification, thinking if I don't eat this right now, I'm going to get something really good afterwards. Okay, now imaging has shown that your inferior frontal gyrus in your prefrontal cortex is involved in resolving interference among competing actions. Okay, it's about cognitive control and delaying rewards. So when you're deciding whether to do something or not do something, so for example whether to go or not go, this cool system, which is your inferior frontal gyrus, is deciding, okay, is it time to go or is it time to not go? Whereas your limbic system, where you find your ventral striatum, is associated with rewards, immediate choices. It's a hot system. So those are your dissociable brain areas, your two separate brain areas to do with long-term choices, which is your inferior frontal gyrus, and immediate choices, which is your ventral striatum. Okay, so there's just a little analogy for you. So your inferior frontal gyrus is like the fat controller. He's deciding whether you get a reward or whether you delay it. So whether you think, okay, stop, hang on, okay, now you can go, or whether not to go at all, or whether to just go straight ahead. And then your ventral striatum is like Thomas the Tank Engine. So it just wants to go, 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 chug along all the time, get that reward, get it right now. Okay. Now, the tasks that they completed in this are called go or no-go tasks, so go, don't go tasks. So there's a couple of examples that we can look at. Okay, so in a go, don't go task or a go, no-go task, you are deciding whether to do something or not do something, whether to go or not go. Okay, so this is just a little example of what one might look like. Okay, so when you see a plain green dot, you're supposed to click on it. If you see a pattern dot, you're supposed to ignore it. Okay, so when you see one stimuli, so a stimuli is like an object that's supposed to arouse something in you. So when you see one stimuli, you are to go, do something, perform a certain action, in this case click. And when you see a different stimuli, you're supposed to ignore it, not go. Okay, so let's have a go. So I'm looking, okay. Oh, plain green dot, so I'm clicking. And there it's measured my time that it took me to make that decision. Okay, I'm looking, plain green dot, click, took me a bit longer that time. See there. So that was a no-go. So you shouldn't have gone on that one. And because I clicked on it, that's incorrect. Whereas that's correct. Okay, again, so that's your no-go stimuli. So not to click. Again, not to click. And not to click. Okay, so then you get some kind of result. Okay, so let's have a look. So there we go. So we've got a table of results. So this would show me where I fall. So you can have a go at this if you want on the PowerPoint and see what your result is and how that fits in with other people. So that's my result there. So I was 92.31% correct because I got one wrong. My score is not the fastest, but not the slowest. Okay, so kind of generally in the middle. All right, so what these tests do is tell you what your ability to control your impulses is like. Okay, so if you can control your impulses, when you got shown that green dot, you wouldn't have clicked on it. I mean, you would have clicked on it if you because your brain is telling you to go, okay, because that's the right thing to do. But when you're shown the pattern dot, if you can control your impulses, you wouldn't go. If you struggle to control your impulses, you would have clicked on it. So sometimes I clicked on it, sometimes I didn't. But there's always a bit of human error. Okay, so in some they use green dots, in some, so in cases study, they use different faces, okay? So it might be that you click on faces that are plain, that show no emotion, but you don't click on ones that do, okay? So other studies have shown that if you do a delay of gratification task in childhood, similar to that marshmallow task, which is a traditional delay of gratification task, then that can predict the efficiency with which the same child will perform on cognitive tasks of impulse control, so go, no-go tasks, as adolescents, so teenagers and young adults. Okay, so this is what the study relates to. Okay, So those who are preschoolers, don't mind scrolling the scrolls all over this, just to help us understand the study a little bit later on. So the other studies show that those who are preschoolers and direct their attention towards the rewarding aspects, so focusing on cookies, which are perhaps the hot aspects, had more difficulty suppressing inappropriate actions, so perhaps not eating them. Okay, so these are known as low delayers. It says there they're not able to resist temptation, whereas the low temptation focus counterparts might have focused on the cooler aspects, and therefore they are called high delayers. They're more able to resist that temptation. Okay, also these strategies could be learned and help children who struggle to resist delayed gratification do so later on. Okay, these findings suggest that performance in these tasks may predict the capacity in adulthood to control your thoughts and your actions. Okay, and the ability to do so can vary by the potency of interfering information. Okay, so in other words, if we can find out if you're a high or low delayer as a child, if you can control your impulses as a child or not, then that also might tell us in an adult how much self-control you have. Okay. So, likewise, a learning or social context can diminish self-control. So, the more realistic something is, the more tempting we might find it. So, we're going to find a face that shows some kind of emotion, like laughter or anger, more alluring, more tempting than a completely neutral face. Okay, so cases study. So the aim of this study was to build on previous research to assess whether delayed gratification in childhood predicts impulse control abilities and sensitivity to alluring or social cues like happy faces at the behavioural and neural level when participants are in their 40s, so they're adults. So in other words, we're getting people as children to do a delayed gratification task, see if they can control their impulses, and then we're measuring them as an adult to see if they can control their impulses or not, and also to see whether that behaviour has a neural correlate, if there's a particular part of the brain which demonstrates that. Now, the alluring qualities of targets in an impulse control task are manipulated to examine behavioural and neural correlates of delayed gratification using an fMRI machine, a functional magnetic resonance imaging machine. Okay, so that's your aim on that slide. Okay, so it's looking at the behavioural aspects, so whether they go or don't go, whether they control their impulses or not, and it's also looking at the neural correlates, so is there a particular part of the brain that is responsible for whether they decide to go or not go. So method, it's a quasi-experiment. Your IV is naturally occurring, so we find out first whether this participant is a high delayer or a low delayer, and that's naturally occurring. We didn't give somebody the position of high delayer or low delayer, we did a test to find that out, so it's naturally occurring. And the dependent variable is their performance on the impulse control tasks. So these are reaction times on the task. So I got measured by my reaction time, also got measured by accuracy. So remember I was 92% accurate. And the performance on these tasks is measured alongside also their imaging results in the second experiment. Okay, so also there is some element of self-report. So the participants complete self-control scales at 2 points in this study, in their 20s and their 30s, and this makes it a repeated measures design. Okay, so this study is a bit complicated because it may seem, well, it's an independent measures design because you're either a high delay or a low delay, not both, and that's kind of right to an extent. However, because they are participating in lots of different tasks, this is counted as a repeated measures design. So it's important that you know that this is a repeated measures design and you can explain why. Okay. So they are looking at doing these self-report scales, age 20 and age 30, and then they're doing some hot tasks and some cool tasks where they're supposed to go or not go, and therefore, because they're repeating things, it is a repeated measures design. Because they're repeating the tasks, it's a repeated measures design. This is a longitudinal study, so it followed participants from the age of 4 into their 20s and their 30s, and some of them it followed until they were in their 40s. Okay. Now, there's quite a lot of information when it comes to sample. So they started off with 562 4-year-old pupils. Okay, these are from Stanford Bing Nursery School. They completed an original delay of gratification task in the late 60s and early 70s, in which they're given a cookie and asked to delay their gratification in order to get the second cookie. Okay, so that's the original sample that we've got. Now, when they're in their 20s, so in the early 1990s, 155 of these were asked to complete, well, all of them were asked to complete the self-control scales, but 155 participants actually did complete the self-control scales. So they rated themselves on their ability to control their impulses. And then again, the same scale was sent out when they were in the 30s, and 135 of these were returned. So you can see the sample is getting smaller and smaller. Then what they did was they wanted to conduct an experiment now. Okay, so there's 2 experiments in this study. The original delay of gratification task, that marshmallow one, doesn't count. That is what is done to categorise the individuals as high or low delayers. So if you're ever asked about experiment 1, that is not what you talk about. Okay, once they've got these 135 people, they look through them because they want people who are above or below average in the original delay of gratification performance and above or below average in their self-report measures. Okay, so they look through these 135 responses to find those people. They find that 117 of them are suitable. So they contact 117 people to ask if they want to take part in experiment 1. 59 of them agreed to take part in the longitudinal behavioural study, so experiment 1. So there's 23 males, 36 females. Yes, you need to know all of this sample. Okay, so if you get asked the experiment 1 sample, it is that 59, 23 males, 36 females of the 117 asked to take part agreed to participate in the longitudinal behavioural study. Okay. So in experiment 1, there was 32 high delayers, 12 were male, 20 were female, and 27 low delayers. 11 were male, 16 were female. Okay, so if you get asked for the sample in experiment 1, all of this information is what you need. All this right here in these last 2 bullet points. Okay. Now in experiment 2, they asked those 59 to come back. 27, so 13 females, 14 females, did. They agreed to be part of a functional neuroimaging study. Okay, now 15 of these were high delays, 5 male, 10 female, and 11 low delays, 7 male, 4 female. So then, one 41-year-old man is excluded from all the analyses because his performance was quite poor. So how they judge whether somebody's performance is poor or not is they calculate the standard deviation, and then if somebody is generally like 2.5 or 2 standard deviations away from the mean, they're considered an abnormality or an anomaly. Okay, so this man was an anomaly. He had an incredibly low score. He was over 2.5 deviations out, so he was removed from the analysis. Okay, so the results for experiment 2 are based on the performance of 26 participants. So we can see here our sample has changed from 562 to 155, 135, 117, 59, 27 and then 26. So this sample has got quite small. So this is called attrition. Okay, so this sample has suffered from attrition. So people have dropped out of our study at each stage. Okay. And some of them have dropped out, so some of them have not taken part, so some of them didn't send the self-control scales, some of them didn't want to come to be a part of experiment 2, some of them experiment 1, some of them were in experiment 1 but didn't want to become part of experiment 2. Now, this might bias the sample in a way. Okay, so we can think about why during next lesson when we evaluate it, but it's worth bearing in mind that this sample might end up being biased. Okay, so in experiment 1, okay, so this is experiment 1 where you've got your 59 participants. Okay, so this tested whether individuals who were less able to delay gratification as children and young adults in their 40s showed less impulse control when given hot and cool cues. Okay, so there's 2 versions of experiment 1. So the first version is a cool version and the second is a hot version. So the 59 participants are doing a go-no-go task. Okay, so in the cool version of the go-no-go task, they're shown neutral faces. Okay, where one sex would be go, that's the target stimulus, and 1 would be no-go, the non-target stimulus. So it might be that I'm shown these images, so neutral male and female faces, and on trial 1, female is go. So every time I see a neutral female face, I'm supposed to click. And then every time I see a male face, that's no-go, that's my non-target, so I'm not supposed to click. Okay, and then trial 2, it might be the opposite or it might be the same. So I'm supposed to click on male faces, but not female faces. Okay. Now, before the onset of each run, a screen appeared indicating which stimulus category served as the target. So a screen would appear saying, on this trial, female is go. Click on female, ignore male, or the other way around. Participants are instructed to respond as quickly and accurately as possible. Each face appeared for 500 milliseconds and it's followed by a 1 second inter-stimulus interval. So it's shown for 500 milliseconds and then there's a 1 second gap before the next photo. Now, you might say that's not very fast, but we're wanting to get instant reactions. We're not wanting to find, when you've looked at that photo and you've thought about it, what is your reaction? We're wanting to see whether you can immediately control that impulse or not. Control the impulse to click. So a total of 160 trials were presented per run in a pseudo-randomized order. Okay, so pseudo-randomized is like on Countdown, which is why there's a picture of Countdown there. It doesn't mean that it's completely random, so it's not like pulling the photos out of a hat, but it means that a computer system, essentially, so a computer formula, like an algorithm, decides which pictures are presented when. Okay, so it's pseudo-randomized. So it's kind of randomized, but randomized by a computer that is using an algorithm. So it's not entirely random because the computer is making a decision, but the researcher isn't making the decision. Okay, so the task was a 2x2 factorial design. So there's the trial type, so whether you're supposed to go or not go, and whether it's male or female. So you're going to have sometimes males where you're supposed to go, so how many times you did, so how accurate you were, and how many times for males you were not supposed to go, so how accurate you were there, or what your reaction time was there. So that's kind of what your table would look like. So we call that a 2x2 factorial design. We measure the accuracy so we measure the accuracy so the percentage correct and the reaction times. Okay, and we've got this in 4 runs, so male, female, and trial type, go, no go. And then the hot version of the task still used male and female faces, but it used faces which were happy or fearful. And this time, happy and fearful served as the stimuli. So happy faces would be go, and fearful faces will be no go, or fearful faces will be go, and happy faces will be no go. Okay, so these tasks were presented using a programmed laptop computer sent to the participant's home. So experiment 1 was done in the participant's own home. If you agree to take part, I'll send you a pre-programmed laptop and you do it on there. Okay. So you have to kind of think, well, how does that impact things like ecological validity? So because you're doing it in your own home, it potentially is more realistic, but then the task isn't very realistic, so it might be lower in mundane realism maybe, but then you've also got more extraneous variables, because, you know, think about if you're doing it and there's a delivery and you got somebody knocking at the door, so that's all going to affect your results. Okay, so in experiment 2 then. So experiment 2, they used an fMRI scanner to analyse those neural correlates. So while you are deciding to go or not go, is there a part of your brain which is related to that decision? So it was anticipated that low delayers would show diminished activity in the right prefrontal cortex. So remember, your prefrontal cortex is where you find your your inferior frontal gyrus, and amplified activity in your ventral striatum compared to high delayers. That's what we think we're going to find. So remember, we've got 27 participants who consented to take part, and they are completing only this time a hot version. So the happy and fearful faces, it's pretty much the same as the hot version in experiment 1. The only difference is the number of trials, the apparatus, and the timing. But the task is generally very similar. Okay, so they're not finding anything from that cool version, so they're just going to use the hot version. Okay, so here's how it was run. So each face stimulus was presented to 500 milliseconds again. And this time though, it's got a jittered inter-trial interval. Okay, so in between each trial, there's going to be a gap, but that gap isn't going to be the same. It's going to range from 2 to 14.5 seconds. And the mean is 5.2 seconds, and that's purely just because of things like the logistics of the fact that you're doing it in an fMRI scanner. You're not just doing it in your own home on a laptop this time. So a total of 48 trials were presented in a pseudo-randomised order again, 35 go, 13 no go, and in total imaging data is recorded for 26 no-go trials and 70 go trials for each expression. Okay, so when you've done 35 go trials, you might find that 35 of those go trials for you are happy faces, and then 35 are for fearful faces, so somebody else is going to do the opposite. So you end up with 70 go altogether for each expression and 26 no-go for each expression. Okay. So the task was viewable by a rear projection screen, and so you're lying in your fMRI scanner. So this is what your fMRI scanner looks like if nobody's seen one. So you lie down and your head goes in it basically. And the task is viewable on this projection screen. So this is where your faces will be displayed, and then you've got a Neuro 5 button response pad. Neuro screen 5 button response pad. So you lie in your fMRI scanner, you press that, that's going to measure your reaction times and also your accuracy. Okay, so one participant was excluded because they were excessively poor on the task. Okay, so we've got 26 results. So this ends up being a 2 by 2 by 2 group linear mixed effects model. Okay, so there was factors of trial type, emotion, and group. Okay, so we're going to have an analysis which is within subjects and between subjects. So within subjects is basically your repeated measures part, and your between subjects is your independent measures part. And again, this is what makes it repeated measures, because you're comparing one person's go score against their no-go score, and then you're also comparing that person's happy score against their fearful score, whether that's go or no-go. And then you're comparing the scores of the high delayers against the scores of the low delayers. Okay, so this is what makes it the repeated measures, this part. Okay, so that's what's known as a 2 by 2 by 2 group linear mixed effects model. Okay, and all of this stuff is becomes important when it comes to statistical analysis and picking stats tests. Okay, so you're going to end up with 8 pieces of information for each. Okay, so what they used in experiment 1, they used an ANOVA. Okay, so experiment 1 uses an ANOVA to do their statistical analysis. Now, an ANOVA, you don't need to know how to conduct, but it's useful to know a bit of information about it. So I'm just going to play a little bit of this video.
This tutorial will talk about
Okay, so this is your within groups part. Okay, so that's kind of what's happening. So we've got this analysis, like you said, of the different groups. So I can there look at their go score, their no-go score, and I can look at their happy score and their fearful score. But then I can also put all of their scores together, and I can look at their score as low delayers, and I could then compare their score to those who are high delayers. Okay, so that's why he's chosen to do an ANOVA there. Why Casey, sorry, rather has chosen to do an ANOVA, because she's trying to find that kind of analysis of variance between groups and as a group. Okay, so you don't really need to know about how to conduct an ANOVA. That's something that you will learn at university if you pick psychology. But what this ANOVA tells us is it gives us a statistic for that data. So you need to be able to analyse those statistics. Okay, so an ANOVA gives us an F value. So here the F value is 2.23. Okay, what we do is we look at that, and we also get given a p-value. Now the p-value symbolises probability, so we'll learn about all this when we do stats tests. But probability is telling us the likelihood that this figure here is significant. So in other words, this is the probability that what we found there is a fluke, is due to chance. Okay, so we're saying that the probability of this being a fluke is less than or equal to 0.1. So in other words, it's less than or equal to 10%. So there's a 10% or less chance that this, what we're finding here in this study, is a bit of a fluke. Okay, so there's 90% chance it isn't a fluke, but 10% chance it is a fluke. Okay. All right, so that was for the effect of the group. So comparing the high delayers and the low delayers, and this is the group and the task. Okay, so you also need to be able to use these significance tables, so you need to be able to interpret those. Okay, and that helps us decide whether it's significant or not. So what this is actually telling us, this figure, but we'll learn how to do that. Okay, now when it comes to reaction time, so remember in experiment 1 they're not inside the scanner, there was no effect of the delay group on the reaction times, on the go trials whatsoever. So writing this out in your notes is an important finding. Okay, and there's some other videos there about stats that you might want to. So it just tells you a little bit more about stats tests, what your p-value is, and what critical values are. Okay, so it's useful in helping you understand these stats tests. So I'd go back to the PowerPoint and I'd watch those videos. So when it comes to accuracy in experiment 1, participants were found to have been highly accurate on all go trials, regardless of whether they were cool or hot. So when they're supposed to click, more often than not, participants do click. Okay, so they've not got 100% correct, but remember there's always going to be some element of human error there. Okay, so they've got 99.8% correct on cool tasks and 99.5% on hot tasks. Okay, so that doesn't matter whether they're a low delayer or a high delayer, they're performed with accuracy on the go trials. So when they're meant to go, they do go. Okay, but with the no-go trials, it is much more variable. So in other words, there's difference between the groups. So the low delayers commit more false alarms than the high delayers. So when you're not meant to click, we're finding that low delayers are clicking more than high delayers when they're not meant to. Okay, so low delayers and high delayers performed comparably on the cool task, but the low delayers trended towards performing more poorly on the hot task than the high delayers. So in other words, on the cool task, we're not finding a very significant difference. It's quite comparable. They're fairly similar. But on the hot task, there is a difference between the high delayers and the low delayers when it comes to that no-go. Okay, so this is why in experiment 2 we didn't bother using that cool task, because it's a waste of our time to repeat that task when we've not found anything, we've not found a difference between the two groups. Okay, so we think that the hot tasks are harder because they're more appealing. So the more appealing something is, if you are a low delayer, the harder it is to resist it. Okay, so overall, therefore, on the go-no-go task, it produced more differences between the delay groups only in the presence of emotional hot cues. So individuals who, as a group, have more difficulty delaying gratification age 4, so low delayers, show more difficulty as adults in suppressing those responses to happy faces than high delayers. Okay, so low delayers have more false alarms. So they couldn't really suppress their ventral striatum. And it could be that your inferior frontal gyrus is more prevalent in high delayers, so it's more active, but we don't know that yet because we've not done the brain scan, but that's what we can assume at this point with what we know about the brain areas. Okay, so that's what the fMRI is for. So what we can assume from this is your high delayers have got more activity in your inferior frontal gyrus, and your low delayers in your ventral striatum. That's what we're assuming this fMRI is now going to tell us. But we're only seeing that on the no-go trials when we're not meant to go, when we're supposed to control our impulses, because on the go trials, we've got comparable results. We're both fairly accurate because both your ventral striatum and your inferior frontal gyrus are going to be telling you to go anyway. So that's why we don't see a difference there. Okay. Now, with experiment 2, we do a paired samples t-test. Okay, so again, I'm just going to show you a bit of a clip of a video about what a t-test is.
Welcome to StatsCast. The purpose of this is to explain
Okay, so this is what this test is showing us. Okay, so obviously we know we have to do stats tests to find out something is significant because looking at a mean isn't enough. But what this test is telling us is looking at between groups, so looking at high delayers versus low delayers. So the variance between those, the difference between the high delayers and the low delayers, comparing it to the variance within groups. So on the go trials and the no-go trials, or on the happy versus fearful faces. Okay, so what essentially this test is telling us is that the two delay groups, so high versus low, did not differ significantly in reaction times on go trials. Okay, so looking at high versus low variance and comparing it on go versus no go, On the go trials, there's no significant difference, okay? So this is an important finding, okay? And they're telling us the probability level again there, okay? Now, accuracy. Overall, accuracy rates for the hot trials for the go, no-go were uniformly high on the go trials. So again, on the go, we're getting like 98.2% correct hits, okay? But on the no-go, we've suddenly got a 12.4 false alarm rate, okay? So now on the no-go, it's time to look at the two groups. So the differences between the two delay groups are consistent with the observed differences in the hot task on experiment 1. So low delayers, just like in experiment 1 on the hot version, are committing more false alarms than the high delayers. Okay, so they're less accurate. And now when it comes to the brain imaging results, okay, so on the brain imaging results, we're finding that go versus no-go trials identify the differential regions of the brain. Okay, so which regions of the brain are to do with cognitive control. So the right inferior frontal gyrus is involved in accurately withholding that response. So it's in control of whether you go or you don't go. So it might be that it's more developed in people who are high delayers. So high delayers show more activity in that right inferior frontal gyrus when actively and accurately withholding a response on a no-go trial. Okay. Now, compared with those high delayers, the low delayers had diminished recruitment of that inferior frontal gyrus. So they're not using it as much as a high delayer is, okay? So there's less activity in that inferior frontal gyrus on a no-go trial, okay? And also they're more likely to make errors. So the ventral striatum demonstrates a significant difference in recruitment between high and low delayers. So this is a reward-related region of the brain, and it's showing a three-way interaction of group and trial and emotion. Okay, so when they're shown a happy no-go trial as a low delayer, there is more activity compared to high delays. So if they're shown a happy face, which they're not meant to click on, and they're a low delayer, they're going to show much more activity in that ventral striatum compared to if they are a high delayer being shown that happy face that they're not meant to click on. So what this is suggesting is that because there's this three-way interaction, it might mean that when the stimuli is really alluring, so we might find happy faces quite alluring, like we want to respond to them in some way, that's something that's hard for us to resist as human beings. So if you are a low delayer, you already struggle to control your impulses. So then when you see that face that's quite alluring and you're being told you can't respond to it, you struggle to do so. So your ventral striatum is quite pushy and wanting to do that, whereas in a high delayer, it isn't. So this shows that the prefrontal cortex differentiates between go and no-go trials to a greater extent in high delayers. Okay, so the ventral striatum showed exaggerated recruitment in low delayers. So ventral striatum more active in low delayers, inferior frontal gyrus more active in high delayers when it comes to no-go trials. Okay, so there's a few conclusions that we can come to there. So the first one, so sensitivity to environmental hot cues like a happy, smiling face plays a significant role in an individual's ability to suppress actions towards alluring cues. So like we saw, if it's a happy face that you're not meant to click on, it's really difficult for some people to control and suppress their impulses. Resistance to temptation, as measured originally by a delay of gratification task, is a relatively stable individual difference, that predicts reliable biases in frontostriatal circuitries that integrate motivational and control processes. So in other words, it's a stable biological difference. So it's an individual difference. It's a difference. There's a difference between you as a high delayer and you as a low delayer, and that is a stable biological difference. So you've got one brain area that's more active when you're trying to control your impulses, and you've got another that's more active when you're being given something that you should control your impulses for. And that is going to be stable throughout your life. So as a child or as an adult, you are going to struggle to control those impulses because that brain area is always going to be the most active for you. That's what it's saying. Okay, the capacity to resist temptation varies by context. The more tempting the choice for the individual, the more predictive are the individual differences in people's ability to regulate behaviour. So if you can't control your impulses, then you've got less activity in that inferior frontal gyrus. So if you are a low delayer, we can essentially predict your ability to resist temptation in future because we know that ventral striatum is going to be pushing for you to get the rewards, basically. Behavioural correlates of delay ability are a function not only of cognitive control but also of the compelling nature of stimuli that must be suppressed. So not just that we can control or can't control, but that because it differs depending on how alluring something is as well. So compared to the happy faces and the fearful faces and the hot stimuli and the cool stimuli. And also individuals who at the age of 4 have difficulty delaying gratification and who continue to show reduced self-control abilities have more difficulty as adults in suppressing responses to positive social cues, so the happy faces, smiling faces, than those who don't. Okay, so quite a few things there. Alright, so there's your reference. So again, you need to be able to learn how to write that reference. And we're going to evaluate this study in the next lesson. Okay, we've already mentioned a few different parts of it, things like types of data gathered you can think about. Also, think, is it a realistic task? Mundane realism, we've mentioned that. Where it's done, the sample, and if that's biased. So I want you to bear all of that in mind because we're going to evaluate it next lesson and recap the study. So this is quite a complex study. You need to make sure that you have got really thorough notes on this study as well as all the background information. I will be checking those notes next lesson before we go on to evaluate. Okay.